Hi, my name is Allison Ellis, and I'm a junior at Purdue University. My research project is entitled Incubation in an Obesogenic Environment Impairs Myotube Reformation Following Cardiotoxin-Induced Damage. How obesity affects skeletal muscle regeneration is an emerging field of study, but its studies in humans are limited. Obesity reduces skeletal muscle quality and the myogenic response to muscle damage. High levels of intermuscular adipose tissue, also known as IMAT, are seen in obese muscle and cause an increase in macrophages, which promote chronic inflammation in skeletal muscle. Previous research indicates impaired regenerative capacity of skeletal muscle in obese mice. Specifically, muscle satellite cell function is impaired. The purpose of my research project is to identify if exposure of primary human myotubes, both obese and lean, to an obesogenic media impairs regeneration following cardiotoxin-induced damage. The methods I used to do my project were satellite cells were isolated from young, lean, and obese subjects. Myotube damage was induced by treatment with cardiotoxin for 1 hour. After this CTX treatment, lean and obese myotubes were covered in an obesogenic, OBGM, or control BSA-GM growth media for 3 days. The OBGM was composed of Oleate, Palmitate, Insulin, and Tumor Necrosis Factor-alpha. Cell viability was assessed by MTT. Myotube fusion and myoblast proliferation following cardiotoxin treatment were determined by immunocytochemistry, and proliferative and myogenic gene expression was determined by PCR. After CTX treatment, cells regenerated in an obesogenic media or BSA-GM for 3 days. Compared to immediately post-CTX treatment, myotube reformation in OBGM only had a 16% increase in proliferation compared to myotubes that reformed in BSA-GM at a 33% increase in proliferation. Additionally, myotubes that reformed in OBGM had a fusion index of 26%, whereas the fusion index of myotubes that recovered in BSA-GM was 35%, thus showing that differentiation of myotubes was lower in OBGM. Additionally, there was no difference in cell viability. OBGM also lowered the myogenic gene expression. For example, the MyoD and MRF4 gene expression in OBGM was decreased by 25% compared to myotubes that expressed MyoD and MRF4 in BSA-GM. Additionally, OB myotubes had an increased myostatin expression compared to lean myotubes. Overall, the incubation of myotubes in an obesogenic media increases CTX-induced damage of primary human myotubes from OB donors. Additionally, obesogenic media incubation also limits the reformation of primary human myotubes from lean and obese donors. These findings could possibly be due to an impaired myogenic gene regulation that I talked about in my previous slide. The limits to my project were that studying the mechanisms of skeletal muscle damage and repair in humans is challenging. In vitro models are not complete representations of in vivo responses. However, there are some future investigations that we could do with this project, such as incorporating gender for how gender impacts skeletal muscle regeneration in individuals with obesity, and how weight loss affects skeletal muscle regeneration in individuals that were formerly obese. Here are my references, and thank you so much for listening to me today. Bye.